Good evening, everybody, and thank you for joining us for this Cafe Sci. My name is Dan Lavoie, and I'm honored to be your host this evening. Cafe Sci is presented by PPG. Cafe Sci is also supported by the good people like you. Thank you to everyone who donates and support this program. If you're unable to donate but would still like to support Cafe Sci, the most helpful thing that you can do is invite your friends to join the audience. Cafe Sci takes place at the first Monday of each month, and you can register for our next month's speaker at carnegiesciencecenter.org. Before I introduce tonight's speaker, I'd like to remind everyone that you can ask questions by using the Q&A button at the bottom of your Zoom window. You can enter your question at any time during the presentation, and we'll get to them at the end, and we'll do a wonderful Q&A session. All right, so it is my pleasure to introduce tonight's speaker, Dr. Amy Welsh. Amy Welsh is a professor at West Virginia University in the Wildlife and Fisheries Resources program, housed in the School of Natural Resources and the Environment. Her area of specialties is conservation genetics, focusing on applying genomic principles to the management and conservation of fish and wildlife populations. Dr. Welsh has been working in the field of conservation genetics since 2001, remaining at the forefront in her research as the field has rapidly evolved and to incorporate new technologies. Her projects have included a wide range of species such as lake sturgeon, candy darter, elk, river otters, king and clapper rails, cheat mountain salamanders, and dragonflies. So, Amy, I'd like to welcome you here, and I'm going to already hand the stage over to you. So, thank you for doing
Okay.
this.
Thanks so much, Dan. I'm excited to be here, and thanks to all of you that are attending live tonight and those of you that watch this recording in the future. So let me go ahead and get my screen up for you all. Okay. So, tonight's talk, we're going to be talking about de-extinction. Super excited about this. So, I have taught a conservation genetics course here at West Virginia University since 2011. And when I started teaching that course, I would talk about de-extinction, bringing species back up from the dead using genetic tools. And I used to just briefly mention it because I thought, oh, this is just such a science fiction fantasy. We're so far from this. This is a crazy idea. And it's been fun to watch my teaching of it evolve over time because it became more and more of a reality. So my classes started spending more and more time talking about de-extinction, the complexities that surround it, the benefits, limitations of it, and because the future is here. So something that I thought was just the science fiction fantasy is actually happening now and it's here today. So it's been very exciting to see how rapidly that has come into fruition. I want you to take note of my subtitle. So, De-extinction, Jurassic Park science, or conservation's new frontier. I want you to keep that question in the back of your head as we go through tonight's webinar. We'll revisit it at the end of the talk, but that's really the core question we're going to be addressing tonight. Is this something that's just a novelty that the media gets really hyped up about? Or does it really truly reflect a new frontier for conservation biology and the ability for us to be able to conserve our endangered and threatened species? So, what we'll go over tonight is we'll go over the history of where we've been in conservation, what de-extinction is, its benefits and limitations. So, hopefully, when we get to the end of the talk, you will have a clear answer for yourself, or probably not too clear, but an answer for yourself about whether or not you think this is Jurassic Park science or actually conservation's new frontier. When folks at the Carnegie Science Center reached out to me about this, they reached out to me about the time when there was news media hype about the dire wolves becoming de-extinct. I got excited, just as excited as everybody else was. It was really, really cool. The dire wolves are just a cool species. Some of you may know them from Game of Thrones, so it was really an exciting time for science, for genetics, for conservation to see this. I was on board. I was very excited to be able to give this talk because again, I was sold on the hype of the dire wolf de-extinction. So we'll be exploring this case a little bit more in depth as we go through tonight's talk about how they actually de-extincted the dire wolves and kind of getting in some of the philosophical questions related to it. Before we begin, though, I want to talk about the field of conservation genetics. So, de-extinction is a part of the field of conservation genetics. It's how I define myself. I consider myself to be a conservation geneticist. So, as I was looking for a good definition of what conservation genetics is, I found my favorite definition on Wikipedia. And basically, I'd like to walk through this definition because I do think it captures very well what conservation genetics is. It's an interdisciplinary subfield of population genetics. So, population genetics is a subdiscipline of the overall field of genetics. It's looking at the genetics at a population scale. And that's essentially what conservation genetics is. It's pulling those concepts and theories from population genetics and applying them in the conservation setting. It is also a subdiscipline of the field of conservation biology. And when I teach my conservation biology class, I stress to my students that conservation biology, although it has biology in the name, it's actually really interdisciplinary. So you need to pull in from a lot of fields in order to effectively do conservation. You need to consider anthropology, you need to consider economics, law, policy, all those kind of disciplines in order to effectively do conservation. And you need to tap into those disciplines when you're doing conservation genetics as well. It's a relatively new field. So, conservation biology really came to its start in the 1970s. If you think about what the legislative outlook looked like in the 1970s, we had a lot of environmental legislation coming on board at that time. We had the Clean Water Act, the Clean Air Act, the Endangered Species Act was enacted in 1973. So, it was a time where people's environmental awareness was at its peak. It was reflected in our legislation, and it was also reflected in our science. So that's when the field of conservation biology developed. And then shortly thereafter, genetics, population geneticists started delving into the field of conservation, creating the field of conservation genetics. As we continue to move through our definition, so we're using this subfield of population genetics to understand the dynamics of genes in a population. So again, our unit that we're looking at is the unit of the population. We're generally looking at how populations are responding genetically. What does the genetic health of a population look like? So we're often looking at things on a population scale and how genes are changing at a population level. For the purpose of, and this is what I particularly like about this definition, for the purpose of natural resource management, although it's called conservation genetics, it's not just restricted to conservation scenarios. So we often use the principles of conservation genetics for general natural resource management, even if it's game species that we're interested in managing. Conservation of genetic diversity. So, the field of conservation biology is trying to conserve biodiversity at multiple scales, including the smallest scale, which is the scale of genetic diversity. And then finally, the prevention of species extinction. That's a key foundational concept in conservation biology that, in general, we are working to prevent species extinction. That can be a little bit controversial because extinction is a natural process. Species naturally go extinct. So, should we actually be preventing all species extinction when it's a natural part of the evolutionary process? However, what we're experiencing today is extinction at a rate that has been much higher in our evolutionary past. So, a lot of people will argue that sometimes we should just be preventing species extinction that are caused by humans. But then the counter argument is that you could argue that indirectly humans are causing most species extinctions. So in general, with conservation genetics and conservation biology in general, we're working to prevent all species extinction. So now that you know what conservation genetics is, giving you that framework within which de-extinction exists, I'd like to walk you through where conservation genetics has been so you can truly appreciate where it's going and where it is now with the current technologies that we're seeing that are being used in de-extinction. So let's go back to the early days of conservation genetics. As I mentioned, conservation genetics came around in about the 1970s. At that time, our genomic technologies were not too great. So they were what we relied on most at that time were a tool called allozymes. Allozymes are not even looking directly at the DNA. So they're looking at protein variants. And oftentimes, what you would do is you would take a sample from an organism, you'd isolate the proteins out of that, and you'd run it through a process called gel electrophoresis. And that's what's pictured here. Basically, what you're doing is you're separating these protein variants, and that's what these different bands represent, the different protein variants that are there. You're separating them out by molecular weight and size. And so you're identifying what protein variants an individual has, and you're indirectly inferring the genetic variation from the variation you're observing at those protein variants or allozymes. One of the biggest drawbacks to allozymes was that it required a very large amount of tissue. You'd essentially have to use an entire organ to get enough tissue to be able to do an allozyme analysis. So that, from a conservation standpoint, really limits you because you would have to kill the organism in order to get enough sample to be able to do an effective allozyme analysis. So if you're trying to conserve the species, you may not want to kill individuals for the sake of gathering genetic data. So a lot of times in the early stages, conservation genetics was rarely used for species of conservation concern because you didn't want to have to kill individuals for research. And so instead, it focused a lot on game species. You can also see on this gel, it looks kind of blobby, pretty difficult to interpret. So there was some level of subjectivity in the interpretation of the data of allozymes. The lab work was pretty intensive. When I was in grad school, we had one lab allozyme project that was still hanging on, working on Pacific salmon, and nobody in the lab knew the technique except my advisor, who was a little bit old, quite a bit older, and knew the old school allozyme technique. So that's when he would pull out all the equipment when that salmon project came through the lab. So allozymes were used in the 1970s to 1980s and again had some serious limitations in terms of their utility for conservation genetics. So it slowed the process of getting genetics into the field of conservation. Things then changed in the 1980s. So in 1983, Carrie Mullis got a Nobel Prize for developing a technique called polymerase chain reaction, abbreviated PCR. And that really revolutionized the field of genetics as a whole, but especially the field of conservation genetics. You can think of PCR as a DNA copy machine. So now you didn't need to sacrifice a whole entire organism just to get so that you can get a whole entire organ to do an allozyme analysis. Now you can just take a very small tissue sample and use PCR to make copies of the genes that you're interested in. This is what revolutionized genetics in the field of forensics, why you can get sufficient DNA from saliva on the end of a cigarette. It's because of PCR. We can make copies of the genes that we want, and now we don't need a lot of starting sample to be able to do our analysis. So this made genetics truly more possible for species of conservation concern because we could more easily get a tissue sample that we could then use for analysis. The genetic marker that we used primarily once PCR came on board was a tool, and we continue to use this to some extent today, a tool called microsatellites. Microsatellites are these short, tandemly repeated sequences of DNA. So, for example, you may have the sequence GATA repeated multiple times right next to each other. And what happens in these regions of the DNA is we see a relatively high mutation rate compared to the rest of the genome. That makes microsatellites really useful when you want to look at contemporary genetic changes in a population. So, for example, if I have a barrier that was created, maybe a dam that was put on the river, and I want to see if there were genetic effects due to that fragmentation of the river and whether that dam is impeding gene flow between the different sides, microsatellites may be able to pick up those genetic differences on a shorter time scale than some other genetic markers because of that relatively high mutation rate. So it's a very useful tool for looking at contemporary changes in a population. The downfall for microsatellites is they're generally considered junk DNA, which means they're not really coding for anything. They're not coding for any proteins, so they don't really affect any traits in an organism. There are some exceptions, but in general, microsatellites are what we consider neutral or non-coding DNA. That means that this marker is useful for looking at gene flow, it's useful for defining population structure, but it's not useful for seeing if a population is able to adapt to environmental changes. For that purpose, you're really going to want to look at genes that are actually coding for something that will likely be under selection. This is an example of what microsatellite data look like. So this is gel electrophoresis, the same process that I described on the previous slide, but a little bit fancier, a little bit cleaner, making it easier to represent. So the different colors represent different genes or positions on the chromosome, and then you can easily figure out what an individual's genetic profile is at multiple locations on multiple chromosomes to develop a comprehensive genetic profile or DNA fingerprint for that individual. So it was much easier to interpret the data compared to Alzheimer's data in the past. Now we are moving from the field of conservation genetics to the field of conservation genomics. So when we were using microsatellites, we may look at only 12 or 15 genes in the whole entire genome, not really a comprehensive sample of an individual's overall genome. With genomics, we're doing a lot better. So now we may sequence the whole entire genome of an organism or a very large random sample of the genome. So we're able to get a lot more genetic information from an individual, giving us a much broader perspective of what's happening in the genomes in a population. So these tools have been around for quite some time. They started off to be really expensive. What we find is that in conservation genetics, we get the trickle down stuff. So a lot of times we'll see the developments in human medicine where there's a lot of money invested and they can afford the more expensive techniques. Usually then trickles down to agriculture, so it gets some exposure to wildlife, and then finally it trickles down to us once the tools become more cost effective. And we're there now. So most conservation geneticists are now conservation genomicists and are looking at that broader picture of the genome in the organisms that they're studying. Because we're looking at more of the genome, we're now able to tap into those adaptive differences that we weren't able to access with microsatellites. So now we can look at genes that actually code for something so that we can evaluate the adaptive potential of a population to be able to respond to any environmental stressors that may come their way. So it really has opened a lot of doors in the field of conservation genetics. I then would like to show you some examples of some projects that have used conservation genetics. So these are all projects that are working on or have worked on in our lab. We'll go up here and start with some elk. So we have an elk project going on in West Virginia. So there used to be an eastern subspecies of elk, and it went extinct, but many states along the East Coast have reintroduced elk. West Virginia has joined that. So we reintroduced elk into the southern part of the state. When we did the reintroduction, we reintroduced two different subspecies. And so one of our projects was looking at genomic differences between those different subspecies and looking at did we sufficiently capture the genetic diversity of the source population. So sometimes when you do reintroductions, if you're reintroducing just a small number of individuals, you may not capture the full genetic diversity of the source population, which can have downstream consequences for your population. The other cool thing we did with the elk is our reintroduced elk were pretty highly susceptible to brainworm. So what we were able to do using the genomics is we were able to identify genes that were associated with brainworm mortality. And once we identified those genes, we could give each individual a brainworm risk score. How at risk were they of getting brainworm? And that can help managers in the state by, if they do future reintroductions, they can then calculate an individual's brainworm risk, genetic risk score, and determine whether or not they should be reintroduced and try to avoid introducing individuals that have a high brainworm risk score. This picture represents a river otter, and I found stole off the web a good picture with it eating a fish because that was the motive of this project. River otters were another species that went locally extinct in West Virginia, and they were reintroduced and are now doing quite well. So well to the point where the fishing community in West Virginia were concerned that the river otters were eating the stock trout. So we were able to use genomics to figure out what the river otters are eating. We were able to analyze the stomach contents of river otters. You can do that using microscopes, but a lot of times you'll miss things because microscopes are just seeing the parts that haven't been digested. Genetics can tap into, usually identifies a lot more species. So I had a grad student who had the fun task of opening a lot of river otter stomachs and doing a genetic analysis to see what species they were eating and try to figure out whether or not they were eating the trout. Turns out they were not eating the trout. Their primary diet consisted of sunfish and crayfish. This is one of my favorite species. This is the candy darter. It is an endangered species here in West Virginia. This is a picture from the Photo Ark. If you're familiar with the Photo Ark project, they came out and photographed the candy darter for the Photo Ark. And this species, the biggest threat to that is hybridization with the introduced variegated darter. So the variegated darter was introduced through live bait bucket introductions. They then have hybridized with the candy darter. And basically, so what we use is genetics. We use the genetics to track hybridization. And pretty much that whole project relies on genetics to be able to accurately to figure out how the hybrid zone is spreading. And the final project I'll highlight is the Cheat Mountain salamander, another federally threatened species. Here we're using genomics to look at population differences in a wildlife refuge, trying to figure out if there are barriers to movement, specifically the effects of snowmobile trails. So snowmobile trails may not seem like much of a barrier, but to something like a salamander, they really could be. So this highlights some of those uses, those traditional uses of conservation genetics, what we've been able to answer with the tools that we have so far. What we are currently missing, and sometimes, and what the technologies that are represented in de-extinction can possibly supply is how do we translate some of our research findings to management in a rapidly changing environment? So sometimes we may come up with these great data, but how do we actually use that for management? And I'm highlighting two examples here in these pictures. The first is climate change. We're seeing a lot of species responding to warmer waters. This is an example of some fish kills. So as waters warm, some fish are less tolerant of those warmer waters than others. There's been a lot of genomic work looking to identify those genes that might make an individual more tolerant of warmer waters. But that's great. Once we identify those genes that give you thermal tolerance, then what? That's the missing link. So how can we actually apply that to management? What can we do to improve the population's adaptive potential to be able to respond to a changing environment? Another example is disease. This is white nose syndrome that's affecting North American bats. So you can see this fungus on the bat's nose. What happens is it'll wake the bat up during hibernation. They'll leave the cave and they'll start looking for food. No food available at that time, and then they end up dying. Um, the the fungus was originally introduced from Europe, and the bats in Europe are resistant to it. So has been genomic work trying to identify those genes that confer resistance to the disease. But once again, once you have that identified, then what? How can we use that information to benefit the population? And that's where some of the tools that are used for de-extinction really might be able to come into play and represent conservation's next frontier. So let's talk about de-extinction. De-extinction, a lot of the technique that they use for de-extinction is based on the science of cloning. Cloning has been around for quite a bit, and you may or may not know, but cloning is actually done quite a bit. There are a lot of cloned animals in the livestock world. Horses, beef cattle, many of them are cloned. In this picture here, every one of these horses is a clone of each other. So basically, the whole horse racing team is a clone of the same horse. So it's done a lot in livestock. If you have a horse that's a good racer, then there's motivation to clone that individual so that you increase the probability you're going to have another one that's a great racer. It's done a lot with beef cattle. You've got a really good beef cow. You want more of that really good beef. Let's clone that individual. It's done with people's pets. So, this is more of a luxury item. This is Barbara Streisand with her cloned dogs. And so it is done with pets. Where, yeah, so people are cloning their pets. So that is an option if you in the audience are considering it. Boy, if you've cloned your pet, I would love to hear about it. That would be really awesome. So it is being done. A lot of us, when we think about cloning, we think about Dolly the sheep, the first animal that was cloned. There were definitely some hiccups in the early days of cloning, but we've actually come a really long way since then. And cloning has been, the technology has improved quite a bit, and it's quite successful. There are things, I mean, there are still problems with some of the cloned animals that do pop up, and sometimes the cloned animal is not an exact replica of the original one. Sometimes it ends up being a little bit different. That can be because there are other things, even though you have an identical genome, which genes are turned on or turned off could be different in the cloned animal. So that gets into the field of epigenetics, where the environment determines what genes get turned on and turned off. So there could be epigenetic differences that cause the cloned animal to look a little different than the original animal. So if you do clone your pet, I cannot guarantee that it's going to be an exact replica of your original pet. When you think of cloning, some people get a little freakish about it, but if you think about it, it is an identical twin that is just separated in time. So identical twins are essentially clones of each other. So you can think of a cloned animal as an identical twin that is separated in time. So the technology, some of the core foundation has been there, and now it's just being applied in a different way. This is a nice diagram showing how cloning is done. So basically what happens is this is the animal, let's say, that we want to create a clone. We take some cells from that animal. So in this diagram, they're taking skin cells from that adult. We take another pig and we remove an unfertilized egg from that female. And then we take out the nucleus from that egg. And essentially, what we're going to do is we're going to replace that nucleus with the nucleus of the animal that we want to clone. And then you use electric pulses to fuse to create that fusion. And then you also generally often use the electric pulses to create an early stage embryo to generate cell division. And then that early stage embryo is then implanted into a surrogate mother who will then give birth to your cloned animal, which will be genetically identical to the original animal, to the donor. So that's the general process of cloning and how it's done. And as I mentioned, the process of de-extinction follows a very similar pathway. So now let's talk about the dire wolf. As we start talking about de-extinction and how we got the dire wolves that we have now today, it's important to travel back in time and let's think about where did the dire wolves live? What was it like back when the dire wolves existed? And I think this is important to understand because I want you, as we talk about the dire wolves, kind of think about what are we actually creating when we're doing this de-extinction. And I think in order to understand and evaluate what we're creating in de-extinction, we have to think about what was it like in the past? What were the conditions within which the dire wolf existed, or the species that you're de-extincting? So they lived back in the Pleistocene. They were found throughout North America. The dire wolf was the most abundant large carnivore at that time. So it was a very common species during the Pleistocene. It coexisted with gray wolves and coyotes. So obviously, gray wolves and coyotes still exist today, but the dire wolves do not. So it's estimated that dire wolves were here in North America at least 250,000 years ago to 13,000 years ago. They went extinct about 10,000 years ago during the Ice Age. So that gives you a general timeline. Why did they go extinct and the gray wolves and the coyotes did not? Probably due to their large size. So they were a highly specialized predator due to their large size. So they ate larger prey. And during that extinction phase, that's what was heavily hit, those larger prey items. So they lost a lot of their prey items. They did not hybridize with the gray wolf, and the gray wolf did hybridize with coyotes. So that could have given the gray wolf a genetic advantage in terms of being able to adapt to the changing climate. And so, because dire wolves didn't have an influx of any new genes, that might have hurt their ability to adapt to the changing climate during that time. So those are our hypotheses as to why they went extinct. Another important consideration is are the differences between the gray wolf and the dire wolf. So they look different. You have clear physical differences. When they looked at the genetics, so they figured out that they last dire wolves and all of our wolves and our wolf-like canids that still exist today, they last had a common ancestor about 5.7 million years ago. So so that's when their last common ancestor was. Then they had all that time to start to genetically diverge. So there's quite a few genetic differences between the dire wolf and the gray wolf. Their common ancestor was a relatively long time ago. That's probably another reason why the two species did not readily hybridize, because they were so, by the time the places were in this era, they had so genetically diverged that they may not have been able to hybridize. So it's important to keep that in mind, that level of genetic distinction between the two species. So that's what it looked like back then. Vicious time, as you can see from the picture. And here we are today. So, the company Colossal Biosciences has de-extincted three dire wolves. Romulus and Remus were the first two that came on board, and their sister Khaleesi was born several months later. It's estimated that these guys could grow to be about 6 feet tall and weigh about 150 pounds. So, again, these are very, very large wolves. They're being held at a facility right now, so an undisclosed facility, and there's no intention of reintroducing these guys back into the wild or into the wild. So there are our new dire wolves. Look like a dire wolf, sound like a dire wolf. And haven't existed on this earth for the last 10,000 years. Pretty cool, pretty exciting. So, how were these guys actually created? How did they generate a dire wolf? And if you want additional details about the process, I would encourage you to check out Colossal Biosciences website. They do a great job of demonstrating how it was done with some cool pictures. But the first thing you have to do is sequencing. You have to figure out, what does my dire wolf genome sequence look like, and then compare it to the most closely related species that currently exists. In this case, is the gray wolf. So what they did is they had various labs that there were 46 dire wolf specimens that were being attempted for sequencing, and only two of those specimens produced usable sequence. It ended up being a tooth that was about 13,000 years old and a skull that was about 72,000 years old. So that alone is pretty amazing and really shows you how far the technology has come when you're able to get usable DNA sequence from specimens that are that old. So they were able to use those two specimens to figure out what the dire wolf sequence of the genome is. The gray wolf is a little bit easier. So there is quite a bit sequence data available for the gray wolf. And when you're working with dog species, that there's a lot known about the canine genome. So not only do we know what the sequence is, but we also know what those genes do. It's what we call well annotated. So we know what the different genes are coding for and what they're responsible for. So they were able to get a well annotated, comprehensive genome for the gray wolf. They can then do an alignment where this is not for the wolves, but just shows you an example of what an alignment looks like. So you take those generated sequences, you line them up, and then you look for differences. Where do the different nucleotides, the different letters in the DNA sequence vary between the two species? You've done that. Now you want to identify what those variants are. But in the case of Colossal Bioscience's approach to de-extinction, they were focused on the traits that are going to make the wolf look like a dire wolf. So if you remember, I said that the two species have diverged 5.7 million years ago. There are quite a few genomic differences between the two species. Instead of changing all those genes, they focused on the genes that were going to give them the traits that make a dire wolf. So the key traits that they were looking for of the dire wolf was the white coat, the larger size, the more powerful shoulders, the wider head, the larger teeth and jaws, more muscular legs, and the characteristic vocalizations that they have. So those were the traits that they were focused on because they felt like if we could edit those genes, then we're going to have something that looks and acts like a dire wolf. So it narrowed it down to about 14 genes and 20 edits, 20 variants that were edited within those 14 genes. So that's what was changed in the genome. Um, just to give you perspective, a wolf has about 19,000 genes in total. A human has about 30,000 genes, just to give you the scope. So they're not changing all 19,000 genes. They're changing the 14 genes that are responsible for the traits that they feel make a dire wolf distinctive from a gray wolf. Once you've identified those variants and you want to make those changes, how do you actually edit the gene, edit the gray wolf genome to have that dire wolf DNA at those 14 genes? The way they do it is through a process called CRISPR. You may have heard of CRISPR. It's oftentimes in the news. It's used for a lot of things in terms of gene editing. Really, another one of those revolutionary technologies that came on the scene that have really changed the face of genetics and what we can do with it. And basically, what happens is you have a guide RNA. So the guide RNA tells this complex, this Cas9 complex, where to go in the genome. What is the gene that you want to edit? So the guide RNA is going to guide this Cas9 to the part of the genome that we want to edit. When it gets there, it's going to bind to the sequence that we want to edit. And then what happens is this Cas9 complex cuts the DNA at that spot. So it's going to cut the DNA. Once it cuts the DNA, your cell flips out. It's like, uh-oh, we've got a nick in the DNA. We've got to repair it. What you have in this reaction is a whole bunch of your dire wolf sequence that you want to put in there. So when it goes to repair that cut in the DNA, it sees all this dire wolf DNA floating around, grabs that, fills that into the hole, and I've now edited my gene so that at that spot, it does not have gray wolf DNA, but instead it has the dire wolf DNA in that spot. And that's essentially how that editing is done. Really cool, really has a lot of potential for a lot of different fields, including conservation. Now you've done that. They've edited the genome so that those 14 genes, it no longer has gray wolf DNA, but it has dire wolf DNA. They take blood. In this case, they took blood from a gray wolf. They isolated, excuse me, the endothelial progenitor cells from that blood. So those are the cells that are in the lining of the blood vessels. Just like with cloning, they remove the nucleus from that cell. They then induced cell division to create an early stage embryo, and then they put that early stage embryo in surrogate moms. Um, for this case, they used the domestic dog. They used hounds because hounds were big enough to accommodate the large pups that dire wolves are larger. The pups are larger than your standard dog. And that's how you get your dire wolf. Pretty cool. So now what I'd like to just talk about, and hopefully you've been thinking about it as we've gone through this process, the different benefits and limitations to this technology. So again, trying to address that question that I brought up on that title slide. So the benefits, a lot can be done with this technology. So even if you're not a fan of de-extinction, the technology alone of cloning individuals and bringing them back has a lot of potential. There is another company that's big on this scene called Revive and Restore. And what this company has done is they have brought back and cloned individuals in order to improve the genetic diversity of the species. So here are two examples. This is Przewalski's horse. Przewalski's horse went extinct in the wild in Asia, so it was it was being kept in captivity. And what happens it was the captive population started losing genetic diversity. Well, there were no other Przewalski's horses to pull from because you had is what you had. So what they ended up doing is they ended up taking DNA cells from a stallion that had died in the 1980s, and that stallion had different genetic diversity than the current population. And they brought that stallion back. So they cloned it and brought it back. And then this is one of those cloned stallions in order to replenish the genetic diversity of this highly endangered population. The black-footed ferret was the first endangered species in the U.S. to be cloned. So Elizabeth Ann was the first one born in 2020. So this is another revive and restore project. Black-footed ferrets in the wild, they got down to 14 individuals, so a very small founding population size. And same problem as with Przewalski's horse. So the population that they're working with had low genetic diversity. They de-extincted, or not de-extincted, but cloned an individual that had died a while ago that had new genetic diversity in order to boost the population. So it's using the same technology that I described with the dire wolves, but it has but a very direct conservation application. What are some of the limitations? So as I've been going through this talk, you might have been thinking of some limitations or some questions that are burning in your mind about this process. Here are some that are commonly brought up. Why are we doing this? Shouldn't we instead be focusing our resources and our money on species that haven't gone extinct yet? So our current endangered species. So instead of trying to bring things back that went extinct, we should be using our time, resources, and money to work to try to conserve our species that are on the brink of extinction. And that's a valid concern. A lot of the stuff that with Revive and Restore and with Colossal, a lot of that is privately funded. And so and then our tax dollars are often going to those conservation efforts. So a lot of times it's different pots of resources that are being used for these purposes. Another concern people have, if we are able to bring a species back from the dead, then are people not going to care if a species goes extinct? Are they going to say, well, who cares if that species goes extinct? We'll just bring them back later when the habitat is better or things get better. Um, as I mentioned, conservation genetics, we are focused at the population scale. And so we have three dire wolves, but three individuals definitely does not represent a population. So how are we going to use this tool to get to that level of what you need for a population? In the case of wolves, a wolf pack generally consists of about 15 to 20 individuals. That's just for one pack. There's been talk about de-extincting the passenger pigeon. If you know anything about the passenger pigeon, there were flocks of hundreds of thousands of birds. That was part of their ecology. So are you going to generate that through de-extinction? So questions of when are we going to be able to meet these population thresholds? And if we just have isolated individuals, have we really truly de-extincted the species? There's questions about, is this really a dire wolf? So, as I mentioned, only 14 genes were changed. There are 19,000 genes in a wolf genome about that. There are more than 14 genes that are different between the dire wolf and the gray wolf. So, is this even really a dire wolf? And then I would argue the extra question of, does it matter? If it looks like a dire wolf, if it acts like a dire wolf, is that good enough? And then I think the answer to that question really depends on what species concept you use. So there are over 20 species concepts out there. If you use a very strict genetic species concept, then you may say, yeah, that's not a dire wolf. But if you use one that's based more on morphology, so the physical traits that we can observe, maybe, yeah, maybe it is. It is a dire wolf. Some people argue they're not living in the same habitat that they once existed in. So if you use what we call the ecological species concept, looking at the niche that that species fills, then maybe it's not the same species. So how do you define a species, and do these de-extinct species fall within whatever definition you're using? And as I mentioned, there aren't any plans to reintroduce dire wolves back into the wild. So some people will argue, then what's the point? And we need to carefully evaluate as we start doing de-extinctions, and this is now a reality, can they even be reintroduced into the wild? There was a paper that came out a little over 10 years ago that came up with some nice criteria for evaluating candidates for de-extinction. So if you're thinking about de-extincting a species and ultimately wanting to release it into the wild, which would probably be the greatest benefit for conservation, these are some things to consider. So first, why did it go extinct in the first place? Have those causes of the declines been addressed? What's the point in doing it if they're just going to go extinct again? Do we have enough information about the biological needs of the species to be able to select an appropriate release area? For example, the dire wolves existed so long ago, we may not really have a good grasp of what their biological needs are. Some of the species that are more recently extinct, like the thylacine, might be one that is more of a candidate for that. And if you do know what they need, is there a suitable release site available? How do these de-extinct species fit within existing policy and legislation? Are we allowed to do this? Would they receive the protection they need so that they don't go extinct again? How do the people in that release area feel about that species coming in? If they have dire wolves coming into their area, they might be concerned about their pets, their children, especially given the size of the dire wolves. What are the ecological, disease, and socioeconomic risks if we reintroduce them? We already have problems with invasive species. Are we just creating another invasive species? And if things go terribly wrong, is it reversible? Will we be able to get those individuals back and say, uh-oh, sorry, we made a mistake. Now we can backtrack. So, in conclusion, going back to my original question, what is de-extinction? Is it Jurassic Park science or conservation's new frontier? It's not going anywhere. So, these pictured here are some candidates for de-extinction that are being pursued. In fact, Colossal Biosciences plans to have a woolly mammoth on the scene by 2028. That's going to be a little bit more complicated than the dire wolf for obvious reasons, but also because they'll need to edit about 85 genes in order to get a woolly mammoth on the stage. So, in my opinion, I think that it's a little bit of both. So, I too get excited about the novelty of this. I find it fascinating that we can do this, and I'm excited by the technology, and I'm excited to have these species back. So it does have that little bit of Jurassic Park feel, which is appropriate given that new Jurassic Park movie came out. But I do also feel like it represents conservation's new frontier. It is a good test of the technology and the limits of that technology and how far we can push that technology, and try and how we can apply it to other things like populations that have lost a lot of genetic diversity, allowing species to adapt to climate change. What if we can edit their genes to give them thermal tolerance or disease resistance? So I think that technology represents a lot for conservation. Whether or not reintroduced de-extinct animals will be a benefit to conservation, it's too soon to tell, but I do think we have to give careful consideration to the guidelines on the previous slide and other such guidelines before we do something like that. So that is it, and I'm happy to address any questions that anybody has.
Fantastic. Thank you so much for being here and doing this. I'm going to go ahead and open up some
Oh,
more
my
questions. We've already
pleasure.
got a few in. Give me a
Okay.
moment here. So we have one question already. I thought Dr. Welsh made a very good point that whether or not it's the same species in quotation, it doesn't really matter. Sorry, doesn't really matter because it looks/sounds like the real thing. With that in mind, do scientists see a specific worth in bringing back ancient extinct species like the direwolf? Do some scientists think we could learn more about a ancient
Hmm.
species, by de-extincting them and then studying the live animals to learn about those ancient species? Or is it more just that it would be pretty cool to see a direwolf or a T-Rex?
Oh yeah, that's a great question. Yeah. And I definitely think that there is a lot of potential to learn about the species. You're right. And so that is an opportunity obviously we'd never had before. So we can hypothesize and guess what a dire wolf was like, but then by doing this, we can really, yeah, it is more than just a novelty. So we could learn a lot about it. There is some trickiness to it, especially the approach that they're using where they are targeting specific genes. So it would be really interesting to see um, if you had the whole direwolf genome and see, because a lot of times genes can code for multiple things, so we may think it's responsible for one thing, but it could also be responsible for other traits too. So it'd be really cool to do that comparison as well. I think one of the challenges of seeing about how it responds is that the ancient species would have had a different microbiome, so microorganisms that existed in it because its diet would have been different. So that could have some impacts on some of the physiological and behavioral differences that might be observed. So some of those things would be hard to get since they can't live in their environment that they lived back then. But it would be quite interesting, and it could help us learn about the existing species, especially species of conservation concern if they're existing today. Yeah, great
Thank you.
question.
That question was brought to us by Pamela Block. We have another question here by Mary Law. For colossal dire wolves, in quotations, and similar cases, given that they are genetically similar enough to gray wolves, would they easily hybridize? If so, would they easily go extinct again due to inbreeding? Would this cause genetic issues for the gray wolf as well?
Yeah. Oh gosh, that's a great question, Marys. Yeah, that is so if they were reintroduced, given that they only have 14 genes that are different, and a lot of the genes are just responsible more for morphology, I would anticipate that they could easily hybridize with the gray wolf. And then we hit a problem, just like you said. So let's say you did reintroduce them and they started hybridizing with the gray wolf. Because of the larger size of the dire wolves, they could potentially outcompete the gray wolf. And you could get an instance where now you've lost the gray wolf because the dire wolf kind of swamped it out. So that is something if we get to the point where we start considering reintroducing these de-extinct species, that is a serious consideration, especially if we take the approach like Colossal did, where you're just editing a relatively small number of their genes. Historically, I think the dire wolf and gray wolf did not hybridize because they were so genetically distinct. But if you're just editing a small number of genes, it could be a problem.
Okay. That's very interesting. I have one question here that I think is also kind of fascinating by Polly Rabinowitz. What if an animal escapes its enclosure and does impact the current environment? Are there existing laws for this? What if the private company creating the animal goes out of business? Who would take control?
Oh, oh yeah. Yeah, those are good questions.
Very Jurassic Park with that. I appreciate
Yeah,
that
very
greatly.
Jurassic Park. Yeah, I like that, Polly. I mean, I don't like it because it would be horrible if it happened, but yeah, it is a very interesting question. So there are laws that prevent it. So there are laws that and and actually I don't know too many details about this because it is that's where it gets gray with de-extinction because these are novelty animals. But generally, I would imagine whatever facility they're keeping them in probably has to follow certain USDA guidelines at a minimum in terms of security and the consequences of those escaping. So I think that there would definitely be legal consequences if they did escape into the landscape. If it went out of business, oh boy, I don't know. I mean, I get my fear is that some person who just is, a big Game of Thrones fan ends up buying the dire wolves, and I don't know what would happen, but that is, I don't know if they ended up having to do any kind of contingency plan for that, and what kind of government approvals they needed to do for this. That's an excellent question, Polly. I would hope that they had to do some kind of government oversight over this and some kind of contingency plan for those kind of scenarios.
Okay. So the next one is a comment, but I think it'll spawn some interesting questions, and I would like to maybe dig in a little bit. And I know your answer to this, has we can have a variety of answers. And the comment was from one of our guests here that says, I don't think that Colossal de-extincted direwolves. It seems to me that Colossal simply selected a few genes, inserted these into the modern wolf question mark host, who gave birth to gray wolves who look like direwolves. And the new
Mhm.
direwolves are white because of Game of Thrones, not their real colors. But I think this is an interesting comment because I think when I see the news and I see conversation, you see comments and social media and other things, that you do get these questions where people have beliefs it's one way or the other. Is there anything you can
Yeah.
give us about clarity about this to help us have an educated answer for those who maybe bring up these kind of questions?
Sure, and I think a spokesperson from Colossal addressed this too, because I think they were after the dire wolves and got all that the de-extinction, they got criticized by that, saying this is exactly what this person has commented. It's not really a dire wolf. And they acknowledge that yeah, that it is not really. It looks like they selected for these traits. It's not as sexy for the media to say that, but yeah, I would agree that it's really you're just picking certain genes to get the traits that you want, but genetically it's still more like a gray wolf. And you're right, they picked. I think the white is because of Game of Thrones, because you look at those artistic representations that I had in my slide, they were not white, they were more like a reddish color. Yeah.
And with
Yeah,
that
yeah.
too,
So yeah.
yeah, go ahead. No, please.
I saw our follow-up comment
Yeah.
and I agree. Yeah. It is I think saying that it's true de-extinction was misleading. And what they had proposed, what other companies have proposed with the passenger pigeon, I was surprised that the dire wolf was the first one to come on the scene. Hadn't really been on my radar when I had been looking at de-extinction. That the dodo bird was a top contender for a while. The passenger pigeon was a top contender. And in those cases, it wasn't that gene editing approach. It was more like the cloning approach. And so then that would be more like true de-extinction. Yeah.
And with that too, that was kind of fascinating too. You mentioned that there's other things you kind of maybe expected to come on the scene earlier. Are there ones that you would expect over the next, say, 1, 5, 10 years that you would expect us to maybe see coming down the road that where people are attempting to do de-extinction or attempting to bring a species back. And especially, maybe I want to focus this is my question on ones where they believe that this could be something that could potentially be reintroduced.
Yeah. I was thinking, so the top for a while when I first got into this, it sounded like the passenger pigeon was going to be the first one. I was skeptical about that because again, the passenger pigeon existed in such large flocks. It relied a lot on the American chestnut, and the American chestnut has been hard hit by disease, and so we don't have a lot of American chestnut around, so the habitat wasn't really there. So I thought the passenger pigeon might be easy from a technique kind of perspective, but from a reintroduction perspective, it didn't seem like a top contender. I was banking on the thylacine or the Tasmanian tiger. So that one is a relatively recent extinction. So I think it went extinct in Australia in about the 19 in Tasmania in about the 1930s. So they had a lot of data about what its habitat looked like. A lot of its habitat is still there. So they had done pretty careful evaluations of the reintroduction potential. So I personally thought that was going to be the first one to go de-extinct. And that could have some positive ecological in terms of where it fit in the ecological in the ecosystem there. So that could have had some positive conservation consequences as well.
Interesting. We'll do one more question here. Pamela Block asked another question. We have a little bit of time left, so we're going to get to this one. A second question, if there's time, which there is. You mentioned that one of the fish in West Virginia is going extinct due to breeding with another species. Is a species considered going extinct when it's breeding with another species and the offspring becomes a hybrid of the two species? Or is this just very broad genetic diversification? I think that's an excellent question and a fantastic one for us
Actually,
to end on.
yeah. That's a fun one, Pamela. So I've been working on the candy darter for quite a bit, and I often think about that because it's tricky. So we often say in conservation, hybridization is bad. Avoid hybridization. Bad, bad, bad. But at the same time, like I mentioned in the talk, hybridization might have been what saved the gray wolves from extinction. So hybridization is also a mechanism for fast evolutionary change in a population. And where it gets tricky with the candy darter is the candy darter does better in cold water. The variegated darter, the introduced one that it's hybridizing with, does better in warm water. Well, we're in a changing climate. That's probably why those hybrids are taking off so well. So we do call it extinction of the candy darter if it's a hybrid. And then over time, what would happen is that hybrid, either the variegated darter will just take over or the hybrid will take over. And given enough evolutionary time, we would consider it's a new species. So hybridization is a way of generating new species. Something we don't often like to talk about in conservation, because again, our usual paradigm is hybridization is bad, bad, bad. But from an evolutionary perspective, I mean, what if that's going to be what saves the candy darter under a warmer climate? I mean, it's a tough one. It's a tough
Yeah.
one. Yeah.
I can imagine we could probably go down this rabbit hole and find a multitude of questions that really split hairs on
Oh, yeah.
bringing species back and conservation and de-extinction. I'll leave you with one last thing here to answer to this. Is there anything in the de-extinction world that you are excited by that is coming down the road? Anything interesting, whether it be new ways, new methods, new research, new development, something that would cause you some excitement in this?
I would say the Przewalski's horse and black-footed ferret examples really give me excitement because that's where I can see direct applications to conservation. And I think where we're getting to next is gene editing of our wild populations. And so species that cannot rapidly adapt to either things like a changing climate or disease, can we give them a boost by gene editing? Now, it does get me a little nervous too, because there are maybe some unintended consequences that we don't know of, but I think people are starting to explore that and starting to see those options, and I think those get me excited. Yeah.
Well, thank you so much, Amy. We appreciate you doing this, and I also appreciate everybody in the audience who was here to listen and provided questions and made this just a wonderful, enriching experience. Give us a few days, and you'll be able to find this on our YouTube page. And if you are someone who's watching this from the YouTube place, please check out some of our other videos. We've had a lot of really exciting guests, and we'll have more to come. So thank you so much, Amy. We really appreciate you being here.
Thank you for having me, and thanks to everyone who attended.
Excellent. You all have a wonderful evening, and we'll see you next week, next month.
Bye.